everyone, welcome to another lecture of drug delivery engineering and principles. So, we are currently talking about hydrogels and we are going to continue that in this class like the last two classes. Let's take a quick recap of what we have learned in the last class itself. So, we have talked about hydrogels, we have talked about the physical hydrogels, which essentially we talked about ionic in this case. Then we talked about chemical hydrogels. We talked about how these are formed. So, essentially what is the major difference between chemical and physical? Physical are essentially molecular chain entanglements, whereas chemical may have a actual bond that is present between these polymer chains. And then we talked about synthesis process. So, typically synthesis process for physical hydrogels involves either heating or change in pH or any other trigger that causes these chains to start interacting with each other and form these gels. Whereas the chemical hydrogels, all you have to do is just add the cross linker or somehow activate the groups. It could be either through light or it could be maybe adding glutaraldehyde or some other kind of cross linker that causes this trigger to happen. Then we talked about some pore size calculations. So, very briefly we discussed this, we are going to discuss this more in future classes. And then we talked about drug diffusion kinetics. So, how can we model how the drug is going to diffuse. So, we found out that we can just use the Fick's law and all we have to really change is the diffusion coefficient D, which will then be affected by whether the gel is microporous or macroporous. So, if it is macroporous, D really does not really have much role to play here, although there are some factors that get added up. Whereas, if it is microporous, then along with these factors we add another partition coefficient Kr, because these drug molecules will now start to interact with that as well. So, let us talk about in situ cross linking hydrogels. These are essentially like this could be any of those previous two hydrogels we talked about, both physical or chemical. But what essentially in situ means is this, that these gels can cross link at the site. So, these are very desirable when you talk about any kind of delivery, because what you can do is you can have individual components, you can mix those two components together, and once you mix those two components together, the trigger is basically given for the polymerization to start, and these things polymerize within a certain amount of time. This could be over a period of seconds or a period of minutes. And so, why it is desirable? Let us say if I am a clinician, I am a doctor and I am trying to inject something through basically a syringe and want to have a hydrogel sort of a system to kind of have this drug release over time. So, all I can do is I can mix the two components together in a syringe and directly just inject into the patient, and what will happen is in that time or in that trigger this thing will polymerize, and wherever it was injected will stay there and sort of form a depot. So, these they are called in situ because they form hydrogel inside the body, and the polymerization trigger could be several types. So, one trigger is time. So, all you have to do is just mix two components together, and it may be like as I said few seconds, 10 seconds, 20 seconds, or it could be minutes, 2 minutes, 3 minutes, and within that the polymerization will start. It could be temperature, right. I mean, I may not even have to do anything in the syringe. So, in syringe it is completely stable, I do not even have to hurry up, but the ambient temperature is let us say 25 degrees Celsius, but when I inject it into the body, our body temperature is 37 degrees Celsius. So, because now there is this change in temperature, something got activated that causes the polymerization, the gelling to happen, and that 25 to 37 degrees Celsius trigger is enough that it then polymerizes, let us say within few seconds. Or it could be pH, let us say and this could be again several types. Let us say in my two tubes that I am mixing together, that combined the pH of the polymer here let us say is pH of 5, at which it does not polymerize. The other tube basically just contains sodium hydroxide let us say with a pH of 8, but when I mix them together that leads to a pH of 7, and at the pH of 7 this thing starts to polymerize. So, this could happen, it could be that I do not even worry about mixing with NOH, I just inject it into the body and our body pH is about 7 anyways, and so when this sorts of diffuse in, it can cause the polymerization to happen. So, any of these things can be explored in if you are talking about in situ gelation. There are some disadvantages to it, obviously there are lots and lots of advantages, but there are some disadvantages. One is this may diffuse throughout the body, right. I mean, if I let us say inject it into an area which is highly fluidic, let us say if it is in my stomach or in my peritoneal cavity or any place which has lots of diffusion and convection going around, then before it can polymerize it can just sort of just diffuse throughout the body. I do not really have a control of shape. So, that is a big problem, because as you know, most of these diffusion kinetics will depend on what is the area through which it is coming out of the system. So, if the area is compact and the drug is only coming out from this much surface area, you will have a different release rate, whereas in case of in situ cross linking, what will happen is once you inject it into the body, it can take whatever weird shape it flows in before it polymerizes, and now you have a much larger surface area and much irregular surface area through which the drug can start to diffuse out, right. In this case it is very hard for me to model how much drug is going to come out in a particular amount of time. So, these are some of the challenges. Another advantage here is obviously if I am looking to fill a defect, let us say it is an injury and some part of my muscle is gone and I want to fill it with a gel containing some cells. All I have to do is just put some liquid over it, the liquid will automatically take the shape. Let us say if this was my muscle and there was some accident through which and this is the shape, then I do not even need to worry about designing the shape. What I can do is I can fill this with the polymer, and once it gels it essentially just takes up the structure that I want. So, there are some advantages to it, but obviously it has to be looked at what sort of applications we are looking at before we can move forward with this. So, again this is very widely used in research, and there is lots of hope for this to get translated to the clinic. And so, we are going to talk about one major research paper on this to kind of understand this. The paper is going to look at different things as well. So, let us get into it. So, in this case this is sort of a paper which is using an in situ cross linking, but then this is here what I am trying to do is basically give you some sort of idea as to how these hydrogels are being used in the system. So, in this particular paper what they are doing is they are using a maleimide based cross linking of a PEG hydrogel to kind of improve the reaction kinetics and the cross linking to happen for both cell encapsulation as well as in situ delivery, right. So, in this case the drug is actually cells, and they want to make a system that they can just inject into the body and not have to worry about surgeries and all. So, in this case the objective of the study is to use a maleimide as an alternative cross linking moiety to polyethylene glycol, and when I say alternative as in try different types of cross linking methods and see which is very widely used. So, before this paper came out people typically were using acrylate or let us say some EDC NHS or some other form of coupling, but now this paper is proposing using a maleimide based cross linking. So, what they have done is they have compared the cross linking moieties, which is again as I said acrylates, diacrylates and vinyl sulfone, which is another functional group. So, maleimide, as we have discussed in the past, is a structure like this in presence of thiols, that this reacts with the sulfitol groups. So, thiols all will have a cetridral, so something like cysteine or any other thiol group might be present due to some other molecule, and this will directly react with this. So, this is again very extensively used in peptide bioconjugate reactions, but then here they are now proposing it to use it with hydrogels. The reaction kinetics is very fast, as you will see in the rest of the data in this paper. This has very high specificity for thiols. So, at physiological pH, which is again for most of the biological applications we are looking at physiological pH. So, this maleimide group has a very high affinity and specificity for thiols. So, it will not have too many reactions going around. So, let us see what they have done in this paper. So, they have used hydrogels. These are again as I said, these are hydrated cross linked polymer networks. They are kind of a synthetic analog to the extracellular matrix through in which the cells typically reside. So, this is a very similar structure as I told in the last two classes, and there is a ease of modification and you can modify them. They will result in some bioactive properties which are independent of what you are encapsulating in them. and in this case they have used PEG hydrogel. Again, as I said, PEG is very widely used. So, and some of the reasons we already know, that it has very low protein adsorption. It is actually used in different areas to kind of prevent the protein adsorption from happening. It's minimally causes any inflammation, although we have talked about some PEG antibodies being formed, but in general, if there is no PEG antibodies, it causes very low inflammation. Safety of use in vivo, again it has been used very widely for last few decades, and there are lots of commercially available products that have used different functionalities to incorporate into them. So, all of these are a plus to this. So, here are some of the methods they have used. As I said, these maleimides will actually react with thiols to form this thioether bond, and what they have done is they have purchased the PEG commercially available, which has the 4 acrylate groups. So, it is a 4 arm PEG. So, here is the cross link between the 4 arms of the PEG. So, this is arm 1, arm 2, arm 3, arm 4, and each of these arms contains a maleimide group at the end. So, and then instead of maleimide, they also have done similar modifications and have got an acrylate and vinyl sulfone, which are 2 of the groups that were used quite a bit at the time when this paper was published. And then again this reaction happens at a pH of 7.4. T is kind of a catalyst to this, and so for acrylate the polymerization happens on the basis of a UV cross linking, and the vinyl sulfone also reacts with thiols to form bonds. And so that is what they have done. They have done reactions, they have PEG and so the reaction that they have done is they have a PEG macromer, they have put an adhesive ligand. What is this required? What do we mean by adhesive ligand? This is a ligand that reacts or that actually interacts with cells and causes the cell to adhere to it. As we said, that PEG does not allow any adherence. So, they have put some adhesive ligands on here that will result in the PEG to be cell compatible. The cells can now bind to it, and so once they do that, they get a structure like this, where let us say at whatever ratio they are making, let us say they are putting this in a ratio of 1:1, then each of these PEG macromer will result on an average will have one of this adhesive ligand, and then they have another peptide cross linker. So, this is kind of a cross linker which contains thiol. So, this one also contains thiol, but it is only a single thiol. So, it can react once, but it will not form a gel, but in this case now they are talking about a peptide which contains thiol at both ends. So, now it can essentially cross link in a whole network like this. So, and they have used different conditions. They have used the adhesive peptide at 4 millimolar or 400 millimolar. They have used it for different times up to an hour, and then the cross linking is done by a peptide which is also cleavable, which is a protease cleavable. So, that means the cells can secrete proteases. So, let us say if the cell wants to move around and it is entrapped in this giant mesh. So, if a cell is here and it wants to move out from here, it can just secrete some protease, degrade this, and this will open up, and then the cell can move out. And so, all of this resulted in this hydrogel structure. As you can see here, they formed it in a disc shape. So, it just took the shape of the disc that they formed it in, and it resulted in a hydrogel. They then let us talk about some of the cross linking chemistries that they have used. So, as I said, the acrylate is a radical polymerization. So, if you have some UV based methods, some photoinitiator is used that will result in polymerization to happen. One of the drawback here is, of course, the UV itself is toxic to cells, and the photoinitiator is also toxic to cells. So, you can actually have reduced cell viability. So, that is a problem that was identified with acrylates for quite a bit of time, and it is not really very conducive to do an in situ delivery, and we will talk about that. It does not really happen very fast. The reaction is very slow, and if the reaction is slow and if you inject it, let us say if it takes 30 minutes, then in 30 minutes all your hydrogel components will get just kind of diffused out into your body, and it really will not form a depot at the site you are injecting it at. And then the other reaction is the Michael type addition, and so this is basically a Michael reaction where a nucleophilic addition happens on another nucleophile. So, this is what they have used to kind of react the maleimide to the thiols. So, again as we said they have incorporated cell adhesive ligands. So, one of the ligands they have incorporated is this RGD molecule. This is a molecule that is very widely found in fibronectin or vitronectin and other ECM molecules, through which these cells have ligands, and when they find RGD in a polymer chain, they can just bind to it and kind of tether themselves from there. So, this is what they have used. They have also modified this to also contain a thiol, so that there is one sulfhydryl group present through which it can react, and then the RGD group is still available for the cells to bind to. So, that is what they have done. So, they have used 20 kDa 4-arm PEG with the thiol containing adhesive peptide, and then in a step 2 what they have done is the degradable polymer, the degradable peptide is this. In this case this is the whole peptide and this VPM site is where the cells will cleave it. So, when the protease are in the solution, they will cleave this VPM peptide once they see it, and then again they modified it with 2 cysteines each containing a thiol. And so, again what that does is that allows you to sort of have a control on a system where now you can have polymerization happening from this as well as it is degradable. So, I hope this is clear. So, now you are talking about, let us say this is our 4-arm chain, one is containing RGD that is capable of binding to a cell, and the other are then cross linked through this VPM. So, if I use another color, so now you have a chain containing VPM from the rest of them, which will then be bonded to another 4-arm PEG. Right. So, that is how the whole structure is formed. So, this is zoomed into one of these, and that is how it is. So, now the cells can actually degrade this VPM moieties to move around as well as bind to this RGD structure. So, here you go, here is just the same thing that I just draw. So, you first react it at a 1:1 ratio. So, you essentially get PEG functionalized with RGD, and then you mix this with the ratio of let us say 1:3. So, now you have 3 sites on which all the VPM gets bonded as well as starts cross linking throughout the network. So, now let us look at what the data they achieve from this. So, first of all, the PEG 4-mal shows higher RGD and VPM incorporation. So, this reaction is more efficient, that is what they are trying to propose. So, let us look at that. So, in cases of PEG 4-mal, what they see is even if they have low amount of the TEA, which is sort of a catalyst here, even at 10 minutes you almost get all the reaction. So, on the y axis you have the unreacted thiol. So, they measure how much of the unreacted thiol is present. So, if they were stoichiometrically mixed, so assuming that all the thiol should have been finished, that is what they see, that almost they get 100% thiol conversion, whereas and the same thing happens at 60 minutes. Whereas, if you look at PEG vinyl sulfone or PEG acrylate, at least for the concentrations of TEA for 10 and 60 minutes, you only see very little incorporation. So, I mean you are talking about only 40% or in this case very little. If now you increase the TEA concentration, which is again a catalyst here, you get to 100 times. So, I mean this is essentially we are talking about 100 times increasing the catalyst concentration. Even then you are seeing not a very good incorporation. Only after quite a bit of time you start seeing almost 100% reaction, and the same thing applies with the PEG 4 acrylate. So, clearly shows that this reaction chemistry is much more efficient to continue with this particular system. Then they look at it, okay, then what is the different polymer concentrations we can use to form this hydrogel. So, what is the lowest possible weight? So, okay, and if you continue to reduce the polymer chains, you may not get a hydrogel at a certain point of time. So, they found that with the PEG-4 even a 3% gel can be formed, whereas with acrylate you can only get a gel at a minimum of 7.5 percentage of this polymer. With PEG vinyl 4 sulfate, PEG 4 vinyl sulfate, you get at 4%, and then the PEG DA is also forming at least at 7.5%. And the gelation times are also very different. So, the PEG 4 forms within 1 to 5 minutes, whereas the other takes up to 30 minutes and longer duration. So, as I said, they are not very conducive for in situ gelation, because if it taking 60 minutes to form a gel, then by the time it is injected in 60 minutes, all the PEG polymer will be all dispersed throughout the body. So, and not only that, if you are encapsulating cells and if you are, let us say, doing it in a cell culture dish, what will happen? Due to gravity, the cells are now settling, and so if let us say if this is your gel 3D structure, let us say if this is your gel 3D structure, all your cells will be kind of populated at the base, and the rest of the gel will have a very sparse population. Because of the gravity, these cells are settling. So, that is why you will not have a very good distribution of the cells either if the gelation time is not fast. Okay, then they went ahead and looked at the hydrogel swelling ratio. So, what they found is that the PEG-4 had a very high swelling at lower percentage. So, if this was the 3%, which is the minimum they can get, and as you increase the polymer concentration, this swelling decreases, whereas in the cases of others you do not really have much swelling happening, because you are requiring quite a high percentages. And so, then they went ahead and encapsulated some cells for 3 days, and here is the data for that. So, what are we looking at now is on these row directions you are seeing each condition, and so if you can see, so the bottommost is the PEG-4 and on this column you are seeing essentially what percentage gel was used. So, this goes from 10 to 4%, and what do you see here is that the PEG DA does not really form gel at the 5 and the 4% as was shown before, like the same case with the PEG 4A. The PEG vinyl sulfone does form gels, but if you look at all these cells, they all look fairly rounded. The only time you are seeing a little bit of an elongation is on this one, but all the rest of them they look fairly rounded. That means when the cells are round, they mean they can't really tether to something. So, when the cells start tethering, they start stretching, they start elongating and spreading onto the surface or onto a gel, but in this case you do not see that. Whereas, if you look at PEG-4 at higher concentrations, yes, the cells are sort of rounded, but as you are decreasing the concentration of the polymer at 4 and 5%, you see them very well elongated. This is how a typical ECM matrix will look like. This is a collagen gel, which is a natural ECM matrix that we find in our body. And so, this is how the cells typically look, and then the only two conditions that are able to mimic that are these two. So, the rest of them are not even able to do any kind of mimicking with that. So, the stains here used are essentially calcine AM, which is a dye that is permeable through the cell membrane, and it goes in, and once it is there it is reduced inside the system to a byproduct which then has a fluorescence. So, only the live cells fluoresce, and then you have a non-viable stain, which is a propadium iodide, which is a dye that is unable to diffuse in if the cells are alive, but if the membrane is compromised, it diffuses in and binds to the DNA and then gives a fluorescent red. So, you can see which one are live and dead. So, all of these are, most of them are showing green, so which means that they are live cells. Then they further went ahead and did some metabolic activity. So, they found that metabolic activity was high in maleimides. So, here you have maleimides in the red, and all of them you see that compared to a 2D control. So, in these are all compared to a 2D control in which there was no gel. You find that they are fairly well metabolically active, whereas the metabolic activity decreases in other conditions. And this is again just a collagen control. Collagen of course has a higher metabolic activity for these cells. These are C2 C12, which are essentially some muscle myoblast cells, and you see that even though the activity is not as good as collagen, but compared to the other synthetic polymer chemistries, they have a much better metabolic activity. And then let us see what happens when you apply this in vivo. So, what these authors have done is they then put it on a mouse heart, so on a live animal. So, this is what happens. So, when you just dispense the little bit of liquid onto these mouse heart, the beating heart, what you see is then the liquid was then spread for a little bit, but then it started polymerizing. So, you get a polymerized gel taking the shape of the external surface of the cardiac wall. And not only that, what the authors have done, they have encapsulated some FITC in here, so which fluoresces green. So, this is all gel, this is all tissue, the cardiac tissue in this case, and what you see is there is some sort of an interface. So, the gel was actually able to diffuse in and polymerize, and then the cells were also able to move in here. So, what you kind of see is sort of an overlapping region. So, you do have this overlapping region in which the cells are interacting with the tissue, and this is a very compatible surface. So, so that is basically all on the in situ hydrogels for this class. We will continue further with our hydrogel discussion and some more computation of the network pore size and all in the future class. Thank you.